Okay, we'll do this asynchronously because of the snow/cold day. So we would have looked at your paper virions today, or I guess it's tomorrow, but I'd like you to bring them on Tuesday. So we're going to finish up a couple of slides on quantifying viral particles and infectious virus. And so we're going to start with the concept of the viral burst. So cells have a finite capacity to produce virus, and that will be characteristic of different kinds of cells and different kinds of viruses. So larger eukaryotic cells can produce as many as 1,000 to 10,000 virions after being infected. The notion of the viral burst and how we measure it in a growth curve was initiated by Delbruck and Ellis, who were studying phage and involved in the development of a synchronous infection model. And that led to the characterization of distinct phases of infection. And you can see them on this slide here. There's an eclipse phase, a latent phase, and then a synthetic phase. And this is a phage graph here for phage growth, and you can see the time for this infectious cycle to really go to completion. You can see up here on the top the total yield per cell. It's basically reached a plateau. That's in minutes for phage, but that's not going to be true for eukaryotic viruses. Okay, so what is happening in this eclipse phase in the early part of infection? If you look here, you're looking at intracellular virus and extracellular virus, intracellular in red and extracellular virus in the blue curve. And during the eclipse phase, no infectious virus is recovered. You basically have what you put in. So what's happening during that phase? What is happening is the uncoding stage of the viral life cycle. The proteins that surround the nucleic acid are being removed, so the nucleic acid can be delivered to whatever compartment it needs to initiate gene expression. The next phase is referred to as the latent phase, where infectious virus can be recovered, but it's only inside the cell. It's not yet released, and that's why you see the gap between the red curve and the blue curve. And then the synthetic phase, as the virus yield increases and virus is being is evident extracellularly. So you could take, for example, the supernatant of the culture. And what these curves look like can be different with the same virus in different cells, or in the same cells with different viruses, mutant viruses. So it can be really, really informative. How do we set up these experiments? Several of you had asked some questions about this for last time. It was on to get you ready for this material was on the prep quiz. So you infect cells if you want to do a one-step. So all of the cells are infected at the same time. You infect cells with enough infectious units so that all or most of the cells are infected at the same time. So usually that's an MOI of 5 or greater. So if I asked you about an MOI of 4 by Poisson's distribution, and there's a slide about that, that would infect a high percentage of the cells, but not all of the cells. So I believe that answer said almost all of the cells would be infected with an MOI of 4, and that is true. So it's probabilistic, and we'll get to that in a few minutes. But basically, you take off the media, you add virus in a small volume, so that the virus can attach. Ultimately, you add media back to dilute away the virus, and then you take samples at various time points. Now, thinking about the MOI, multiplicity of infection, why couldn't you infect with one infectious particle per cell to get all of the cells infected? Why couldn't you use an MOI of 1? And the answer to that lies in probability. Think about the cups in that game on the midway where you're throwing the balls and trying to land them in the cup. Sometimes a cup will get 2 balls, sometimes a cup will get no balls, and there's a good blog post from Vince Racaniello that talks about multiplicity of infection, or MOI, and I've linked to that from the Canvas site. So you should look at that, and you should read about Poisson's distribution. And I don't really care that you can use the formula, it's complicated, but I care that you understand the concept that it's probability, and that you need to be beyond a certain point to really, and that it depends upon the number of cells in the infection, to understand if you're going to have uninfected cells, or what the probability is that all of the cells you have will be infected. Now, small volume for your infection is important because it means that because viruses aren't motile, it increases the probability that the virus and the cells are going to touch each other. If you just put the virus in the media and it was sort of viruses were moving around by diffusion, they would have to randomly bump into a cell. So typically you take the media off, you leave just a little film of media to keep the cells happy, and you add your virus and you sort of rock those cultures so that the media with the virus has a chance to make contact with the cells. We do this in the cold at 4 degrees. Why do we do that? If you were with me, I'd elicit some answers. But it has to do with membrane fluidity. So if you did your infection at physiological temperatures and the virus attached to its receptor, it would go in. And then maybe in another part of the cell, another one would go in a little bit later. So without halting viral entry by keeping the membrane not fluid, cold at 4 degrees, it wouldn't be synchronous. It would be things would be sort of happening in an ongoing manner. So at 4 degrees, when membranes aren't fluid, the viruses attach, but they just stack up there on the membrane. And then when you decide that the adsorption, not absorption, adsorption period is over, typically you add fresh, warm media. And what that does is dilute the virus, and all the virions that are stacked up on the membrane when it gets warm will go in because the membranes will warm up. Okay, does that make sense? You can answer me. And then you take samples at various times post-infection. In my lab, we would typically take one right when we started, and we would call that our time 0 to make sure that we actually added what we thought we added. And then at various times, and then you would assay them by plaque assay. So, multiplicity of infection is the number of infectious units, typically that's how people set them up, added per cell in an infection. But you could also set up an infection using particle number if you wanted to. And remember, we talked about how those two things aren't the same. So, multiplicity of infection that term doesn't mean the amount of virus to ensure that all cells are infected, or it's not even the number of infectious particles each cell receives. So, for example, if you added 10 to the 7th virions to 10 to the 6th cells for an MOI of 10, every cell doesn't receive 10 virions because of that tennis ball into the cup game. Some cells get more, some cells get less. And so you need to read about the Poisson distribution. So why is this important? The Poisson distribution is relevant to a one-step growth experiment because a one-step, if you really want it to be single cycle, requires that all of the cells are infected at the same time. So if you infect with an MOI of 1, only statistically, only 37% of the cells are infected. At an MOI of 3, 95% of the cells are infected. At an MOI of 4, more of the cells are infected, but there's still uninfected cells. So it's important to understand, to really get a grip on what this table looks like. The other thing with MOI is that, often students misinterpret multiplicity and think that it means that it has to be all of the cells are infected. You, as an experimentalist, get to choose. So you can do a low MOI infection, and in a low MOI infection, it takes several cycles of replication before all the cells in the culture are infected. Look at the graph on the right. It's not one step. You infect some of the cells, those burst, and then uninfected cells in the culture can be infected with those released viruses. And so low MOI infections are useful experimentally if you're looking at viruses that might have a difference in their replication kinetics or the ability to be released from this cell, or if they differ in induction of an antiviral state. We'll talk about that later in the term. But you get to choose as an experimentalist. High MOI infections, another point that you should consider is once you reach the stage where you're infecting all of the cells, if you infected with more, it doesn't necessarily mean that you're going to get a larger burst, again, because the cell has a finite capacity to release virus. And in fact, maybe infecting with more would induce a higher antiviral response. You might reach a stage where you get less virus by adding more PFUs. So that's a point you should consider. Now, if we were together, we would do a clicker question, and I would ask you if you think that high MOI infections are physiologically relevant. I want you to think about it, and we'll talk about it again on Tuesday. All right, so now we're going to move on into the information about genomes, genetics, and structure. And there are some slides in the slide deck here that, because we're not live, I'm going to sort of point to them so that you can look more on your own time. Okay, so the viral genome is the nucleic acid-based repository of information so that a virus can build more of itself, replicate, and be transmitted. And in a number of cases, and there are lots of different kinds of genomes, and we're going to focus on a handful in this class as examples. In a number of cases, you can initiate an infection just by transfecting a purified genome. And one of the questions on the final exam in this class is going to be whether or not a particular kind of genome is infectious. So that's a question that ends up being important. And in order to understand the answer to that, we began talking about that on the first day of class when I did that new exercise this year where I asked you to think about a positive strand RNA genome and a negative strand RNA genome. So if you initiate infection with a positive strand, I would ask this again, and I ask you to think about the answer. The DNA that serves as a template strand for messenger RNA is one of these. So think about that. Okay, so we talked about the importance of messenger RNA and viral genomes encoding messenger RNA that will encode viral proteins. But viral genomes do more than simply encode viral proteins, and it's important to think about what else they do. They serve as templates for progeny genomes, and I'm introducing here the concept of the genome and the antigenome, and you can see the direction of those arrows. If a virus had a negative sense RNA genome, and we draw that as the arrow pointing not in the message sense, then its antigenome, which would be the template for more genomes, would be positive sense because the baby genomes are negative sense. Okay, so there are signals in the genome for replication, right? So that replication starts and stops at the right place. So what else do viral genomes do or not do? They serve as templates for antigenomes. They encode, some of them encode proteins. They can be decoded, maybe through several steps to encode proteins. So they encode genes, products, and regulatory signals that are required for replication, as I just said, for packaging, so that packaging of the genome in new progeny virions. You wouldn't want to package non-viral material. They have signals that are important sometimes for timing the replication cycle, sometimes signals for evading host defenses. So lots of things. And so I'm introducing you to tables and figures in chapter 3 that pull all of this together. So this one that's on this slide shows you where the packaging signals are for retroviruses. And for example, we're going to talk about how retroviruses are packaged, how retroviral messenger RNA, positive sense messenger RNA, is packaged into progeny virions through interactions with this packaging signal. So people study those. What's not contained in viral genomes? Not genes encoding protein synthesis machinery. So the viruses are dependent on the host translational apparatus for protein synthesis, but some viruses encode RNAs and proteins that play a role in that process. They tend not to encode proteins for energy metabolism or membrane biosynthesis, but that may actually turn out not to be true as we consider some of the giant viruses. But we're not gonna spend much time on those. We're not going to spend much time on the exceptions in this class because we want you to sort of get what the rules are. And they don't have classical telomeres. So I want you to start to use chapter 3. It includes boxes with genome configuration, gene expression strategies, information about dependence on the host, and it's really, really useful. And these images on this slide here aren't necessarily understandable to you right now, but they will be when we get into the molecular biology. And so this is going to be a good place to go back and look at in order to, when you're studying, for example, for the final. So here's the positive sense RNA genome of picornavirus, for example. The first thing that happens is the generation of protein. Some of that protein is used to be protein for a virus particle, but some of it is also used to generate a negative sense antigenome, which is the template for the generation of more message sense, positive sense, genome sense RNA that gets packaged and serves as genome. So that's what this upper left-hand diagram looks like. And there are these for other viruses that we're going to use as our examples. So we're going to use picornaviruses, we're going to use retroviruses, and we're going to use influenza virus, an orthomyxovirus, and rhabdovirus, rabies or VSV, as our examples. So a big question in this class is going to be, how is the genome decoded to give rise to messenger RNA? What are the gene expression strategies of viruses? How are the genomes replicated? And to what extent and for what steps is replication dependent upon the host? And knowing what the genome structure is, you should be able to just deduce those things out of first principles. We draw genomes as, straight lines, arrows in our class, but that's not what they look like. So this figure in your textbook in Chapter 3 shows you something about what the viral genomes look like in the virus and structures there that are critical for its function. So the topology of double-stranded DNA, for example, has major implications for replication. So this is a great chapter to go back to rather than sort of digging through another chapter to try to find what was that thing that it said about picornaviruses. The other thing that's really critical, and I alluded to it in the last class, is that for viruses, compact genetic material, one gene doesn't necessarily mean one messenger RNA. And so this figure gives you a sense of, for example, a nested set of related RNAs that might have different untranslated regions, different translational start sites. So it's why I harp on the fact that it doesn't make sense to talk about an untranslated region in DNA because DNA isn't translated, and because one gene that you would see in DNA could template multiple related messenger RNAs, each with a distinct untranslated region. And so it's just more precise to talk about an untranslated region on the molecule, which is messenger RNA, that gets translated. It's just more precise. And so again, this figure here is going to be really useful for you when you start to sort of put all the things together from this class. Okay, so that was sort of like the tour through those figures in the chapter. And I want to spend a couple of minutes and talk about genetics, how we use genetics as virologists. And so there's classical genetics, viral mutants that one can select in the lab, or that are selected in a host, or that are selected in nature. And then there's sort of modern molecular genetics based upon recombinant DNA technology. And so this chapter 3, covers an introduction to viral genetics that's used throughout the rest of the course. So there are, because viruses have polymerases that have error rates, there are spontaneous mutations that arise during replication, and one can induce mutations as well. And so you can screen virus stocks and select, identify conditions that would enrich for viruses that have phenotypes that you might be interested in. So viruses that spread better might have larger plaque sizes. Viruses might have the ability to replicate in different cells. They might be resistant to certain drugs, and you could look at those. And so this plaque assay that you're seeing down at the bottom right is an adenovirus small plaque mutant, the one in the second well on the top row of wells, that has restricted spread relative to the wild type, which is Ad5 right next to it. And then this was subjected, that virus was subjected to random mutagenesis with ultraviolet light, and then 27 large plaque mutants were isolated, and the vast majority of them had lesions in a particular gene and in a particular region of that gene. And so this is how you can use viral genetics, classical genetics, to understand what about the viral protein E1B? How does that play a role in spread? You can understand the molecular and cellular biology, and even in a regular stock of virus that you might be titering, you might see small plaque or large plaque variants, and so it just pays to be alert when you're scoring plaque assays, counting plaque assays. Okay, the mutation, which are mutations, changes in DNA or RNA, can be base changes or insertions or deletions or rearrangements. They can be nonsense and missense, but we talk about mutations in nucleic acid. That term shouldn't be used to describe changes in proteins. We can get those through chemical treatments or mutagens, UV, and for RNA viruses, there's a high percentage of mutants. And you can look at in the textbook to find the answer to this question. What is the mutation frequency? What is the mutation frequency in DNA viruses? So for RNA viruses, 1 in 10 to the 4 or 10 to the 5th. So one would say that the frequency was 10 to the minus 4, which is the same as 1 in 10 to the 4, right? And so for DNA viruses, the mutation frequency is 1 in 10 to the 8th to 10 to the 11th. So much, much more mutation, higher rate of mutation for RNA viruses. RNA genome. That genome is going to make proteins. And usually one of those proteins is an RNA-dependent RNA polymerase, and then you're off to the races. Okay, so let's talk about how one could use molecular genetics to study viruses. And that's to engineer mutants, specific mutations. And so for double-stranded DNA viruses, this is pretty easy. One can take linear fragments of viral DNA, shown in the drawing that I've made on the right, and a mutant virus that has a mutation in a particular gene. We put an X in this diagram, and then introduce those pieces of DNA, the linear fragment of viral DNA, and the double-stranded, in this case, circular DNA genome, plasmid, and introduce those into cells, and recombination, homologous recombination, if there's homology on the DNA fragment that spans the mutation site, will generate wild-type virus. And this is called marker rescue. It rescues the virus from the mutation and gives a wild-type phenotype. And this approach, back in the 70s, allowed mutations to be mapped to specific portions of the physical map of the viral genome. You can also do it sort of backwards to introduce mutations into wild-type DNA virus genomes, and you can imagine that using modern technology, you could synthesize a fragment that had homology to the genome and that incorporated a particular mutation you wanted, and you could basically force that recombination. These days, you can even do that using constructs with bacterial origins, allowing the recombination to happen in bacteria, and then screening the resultant fragments, resultant plasmids. All right, so I'm going to give you an example of how that was done, a system that was generated to do that really easily in herpes simplex virus infected cells in my husband Steve Rice's lab. And so one of the genes that he's particularly interested in is the gene for ICP, infected cell protein 27. And so you can see in this image that maps to a certain part of the viral genome. The genome is shown up on top. And what he did was make a piece of plasmid DNA that had homology to the ICP27 locus on the 5' and 3' ends of this piece of plasmid DNA. But in place of the ICP 27 gene coding sequences, he introduced the gene for beta-galactosidase, the LAC-Z gene. And so with a specific substrate, beta-galactosidase can turn the plaques blue. And so what he did was he co-transfected this piece of plasmid DNA with the LAC-Z gene in frame with the coding sequence, the promoter of ICP27, and complement and allowed that to recombine. And he did it in cells that are called complementing cells, complementing V27 cells. And he did that because he was trying to make a mutant virus that lacked ICP27, that had LAC-Z in place of ICP27. And ICP27 is essential for replication. So he needed to provide the ICP27 gene product in trans, right, in those cells. And so these cells express ICP27 from a different expression plasmid when they are infected with herpes viruses. And so he allowed this recombination to take place in these V27 cells, and he picked the plaques, you can see on the left, that are blue. And so he generated this very useful virus that was deleted for the gene that he cares about the most, which is ICP27. And he called that D27 LAC-Z.
Using the LAC-Z virus, the D27 virus that has LAC-Z in place of the ICP27 gene, the same thing can be done in reverse, generating or using a mutant ICP27 plasmid with homology to the locus, and then basically forcing the recombination of the plasmid sequence into the viral genome. And again, that gets done in complementing V27 cells. And in this case, one can pick the clear plaques, and those should have replaced the beta-galactosidase gene. So this is a system that can be used for many double-stranded DNA viruses, a similar kind of system, to generate deletion mutants and then generate specific mutants with engineered sites. I want to end this genetics part of the lecture with asking you to remember that recombination, which we just talked about, is different than complementation. A non-genetic interaction between viruses can happen in cells that are co-infected with two different viruses. And so that's when the gene products of two different viruses interact in the context of the cell to functionally complement each other if the mutations are in different genes, thus permitting virus replication. So it's important to distinguish complementation, which occurs at the level of proteins, from recombination, which occurs at the level of the genome. So in complementation, if the mutations are in separate genes, each virus supplies a functional gene product and the virus can grow. If the viruses carry mutations in the same genes, they're both defective, then you wouldn't get viral growth. All right, so we're going to move on.
If you go
on And here, what I would do is give you a very, very quick quiz looking at ratios of virus to cells and asking you to determine the multiplicity of infection. And so I'm going to give you those now. I'm going to just run through them very quickly. You can think about what the answers would be. All right, so
Up to
in this super quick quiz, look at the diagrams and determine the multiplicity of infection. You can assume that each virus is infectious here, and really we just covered this a couple of minutes ago. So in this, the viruses are blue, the cells are in a dish, and they're yellow, and I'm asking you what's the multiplicity of infection, and remember, it's the ratio of PFU viruses per cell. Okay, this is going to be speedy. Ready, go.
number 2.
What's the MOI? One virus, 10 cells. This one's easy. What's the MOI? It's a ratio, right? And the units are PFU per cell. How about this one? I've even counted for you. What's the MOI? Here's another one. Again, I'm so good I counted for you. Okay, how about this one? It's a ratio.
Both.
It's a ratio. All right, so we're going to move on. Thanks for playing along. No points for that, but you get the idea. We're going to talk about viral structure, and it reminds me of architecture, and so I start with this picture. One thing that I want to tell everybody is that the prep quizzes that I give you on Canvas are to, make sure that you are looking at the slide set before class, to incentivize that behavior, to incentivize maybe looking in the textbook a little bit, but it's obviously your choice, and to get you ready because you've already begun to process some things, to get you ready for class. I, don't necessarily expect that everybody is going to get every answer. So please don't ask questions about why you thought something was right and something was not marked right on the quiz before we have class, because my whole point in doing that is I'm going to talk about those things and you will be more ready to receive them and to get that information if you've done the quiz, and probably particularly if you got something wrong. So I think this material is challenging. The text can be challenging. And so class adds value. It's my role is that of interpreter. And so wait until we have class before you ask questions about a particular prep quiz, because my hope is that all of those will be answered by the end of class. And if you have a question content after class, absolutely write to the Canvas, to the discussion board, because that will make class a richer place if people are willing to do that. But wait until you've had a chance to look at the material in a preparatory way, do the prep quiz, come to class, listen to me, ask questions in class, and then maybe go back and look at the textbook if things are still confusing, and then ask. Okay, so let's do that process and ask on Canvas or in class. All right, so now we're going to talk about viral structure and look at some pictures of beautiful viruses.
Negative strand, single-stranded RNA viruses like rabies.
Okay, so let's talk about the functions of the virion particle, the structure. Virion particles are responsible for transmitting the genome within them from one host cell to another, either within a single host as the infection spreads from the initially infected cell, or among host organisms as the virus is transmitted. And so that presents some paradoxical requirements that are interesting. On the one hand, the virion has to protect the genome out in the environment. Think of environments such as sewage through which viruses get transmitted. But on the other hand, when the virus attaches to a host cell that has the appropriate receptors, the virus needs to then disassemble and deliver the genome to the interior of the cell. So the virion has to be stable but ready to disassemble and capable of disassembling in the right place at the right time, not prematurely in the environment. The virion also needs to have signals on its proteins that allow for critical enzymes, for the genome, and for other protein factors to be packaged within that structure. And so what's on the bottom of this slide is a series of images of cryo-electron microscopy of the virus that I have worked on, reovirus, mammalian reoviruses. And on the left-hand side, you see the environmentally stable virion. In the middle, you see what happens after that virion attaches to a susceptible cell and begins the process of disassembly. It's the intermediate subvirion particle. This is the particle that can penetrate membranes. And then what it delivers to the cytoplasm is the viral core, and that core becomes transcriptionally active, and you can see the vertices have opened up, and it's through those vertices that the messenger RNA gets extruded after the machinery at the vertices becomes activated, and we'll talk about that a little bit more. It's fascinating.
Herpes virus, VSV, influenza virus.
Okay, so virions are metastable structures. They've not attained their minimum free energy conformation, and this is really important. Contact with the receptor for some viruses and or exposure to the appropriate intracellular environment, and we can talk about what that might be, what those conditions might be, can trigger the substantial, irreversible conformational changes in the virion. So we're going to talk a lot about what those triggers might be and who the receptors are, but I think it's important to recognize that virions essentially function like molecular machines that really play quite an active role in delivery of the genome. They are metastable. And just to give you an idea of what that might look like, this slide shows an example of a conformational change. This is the influenza virus hemagglutinin, the H of H1, or H3, or H5. Then there's another molecule on the surface that is on the virus surface that is the neuraminidase, and it gives you the H some number, N some number description of those viruses. So on the top is the cell surface, and you can see in the left-hand diagram, the hemagglutinin is binding as a trimer to the surface of the cell-specific cellular receptors, and in a particular place at a particular time, that hemagglutinin undergoes a dramatic, well-characterized by X-ray crystallography molecular change. The heads move and fall down. Things happen in the core of the trimer, and those changes bring those two membranes together and result in membrane fusion. It's really interesting, and we'll talk about it a lot when you guys do the description of one of the 4 viruses that we're going to talk about next week. So this is primed. The conditions that prime this change are the acidic pH in the lysosome, endolysosomal pathway. More on this later.
Right-hand side, reovirus.
Okay, terminology is important. Look at that material in the textbook and use the right words. It's capsid, not capsule, for example. Okay, so now we're going to go on and we're going to talk about methods to study virus structure. And what I really want you to appreciate here is how beautiful these are and how their morphology, it's a word that somebody was talking about in the last class. I think it might have been Nathan, or maybe it was Timmy. How beautiful morphology is.
Virus, rotavirus.
Okay, so this is using the approach of electron microscopy with negative staining. And so you can see what the resolution is, and we know that biological materials don't have inherent contrast. And so EM with negative staining uses a stain, often heavy metal, to provide the contrast. And what this allows you to do at a resolution of about 50 to 75 angstroms is to visualize morphologic units, things that protrude from the surface of the virion. Sometimes we call some of those capsomers. This is not giving you detailed structural knowledge, but you can see that the viruses differ dramatically. So influenza virus has this sort of amorphous, polymorphous shape. They don't all look the same. That's not true, and that's an enveloped negative strand RNA virus. That's not true of rotavirus or papillomaviruses or adenoviruses, which are icosahedral viruses, and they have a very defined structure, and they really do look the same generally from one to the next. That is not true of influenza viruses.
Virus, both of which have.
Sort of next down in the resolution, this is the inside. This is a cutaway view of the inside of a reovirus particle. And so you'll remember that I showed you on a previous slide what the transcriptionally active core looked like. That's what's delivered into the cytoplasm. Remember that these viruses have a double-stranded RNA genome. That genome stays when the core gets delivered to the cytoplasm inside the virion, and transcription happens through viral proteins that are organized just under those vertices, sort of the red things, and those are the molecules that convert. They don't actually convert. They decode the template strand of the double-stranded RNA genome segment into messenger RNA, which comes out then the portal at the sort of aqua vertices. It's amazing. And there is a different double-stranded RNA genome segment. This is a segmented virus at each of those vertices. Fascinating. That's my virus. This is a bacteria. This is a phage reconstruction, and it's here to show you that really this approach is getting more and more refined. This one is it's actually a non-icosahedral virus. This is an electron cryotomograph, cryo-ET, of a herpes simplex virus virion in A, a vaccinia virus virion in B, and HIV in C, just to show you the kind of image reconstruction that computers can do. Yes, a resolution. Next, the next level up with respect to resolution is X-ray crystallography. And here we're down to low single digits in angstroms. For really complicated large viruses, X-ray crystallography has its limits, at least right now, but you can crystallize component proteins of more complicated viruses. So on the upper right of this slide, there's a small icosahedral virus, tomato bushy stunt virus, for which we have an X-ray crystallography solution. And then on the bottom left is one of the proteins, the outermost capsid protein of the revirus particle. And here on the next solution, you can see all three. You can see the atomic structure, you can see the cryo-EM with image reconstruction, and you can see a negative stain EM. So you get a sense by looking at the slide of the different levels of resolution. So now let's talk a little bit about symmetry. We've been using those words for viruses that have small genomes. That they have to demonstrate genetic economy, and so their protein shells are built by using the same type of molecule over and over and over again. And viral proteins that make the components of capsids have structural properties that permit repetitive interactions between them. And so there are two kinds of symmetry that we'll talk about. Helical symmetry, which you can see on the left. The genome is the dark blue inside, and then there's a repeated protein that winds around that, interacts with the genome, and winds around it in a helix. And then on the right-hand side is an image of an icosahedron. Now at around. So let's talk about helical capsids first. Helical capsids are open, and so it's interesting because then any volume can be enclosed by varying, the length of the helix. So if the nucleic acid molecule was longer, the helix would just be longer, shorter. What's interesting is that all enveloped negative strand RNA viruses, we're talking influenza virus, we're talking the rhabdoviruses like rabies, you can see, or VSV virus, vesicular stomatitis virus down at the bottom. All of these have helical nucleocapsids, but they don't all look the same. So you can see tobacco mosaic virus in a second panel there on the right, what it looks like, and what its nucleocapsids look like. And then you can see vesicular stomatitis virus, which looks more like a bullet. So the size and the interactions among those nucleocapsid proteins that surround the genome may be different, may be larger, may be smaller, but they have repetitive interactions that allow them to interact with each other as well as interacting with the genome. 4 to 5 angstroms is icosahedral capsids are fundamentally different. They are closed structures with a fixed internal volume. So you couldn't engineer a virus that had probably twice the amount of nucleic acid and get it packaged because it wouldn't fit inside the virion. And these icosahedrals are made up of 60, is the smallest number, of identical subunits, and those are the they have 20 triangular faces. You can see those are the purple triangles up on the top. 12 vertices, and those vertices are related by 2, 3, and 5-fold axes of symmetry. And so each of the 60 subunits in a simple icosahedron interact with their neighbors in an identical manner. And so you can see here, this right here is the 5-fold axis of symmetry. It's a vertex. Here's one, here's one, here's one, here's one. I'm not sure you can't see my arrow. Cryo-electron microscopy. Okay, so I was taking virology before the Civil War. I could understand this when my professor was standing right in front of me, and then when I was on my own, I had a really hard time doing it. So I understand perhaps your frustration with, okay, but I want you to be able to find axes of symmetry. And so we might have to do this together because it's hard for this to be translated appropriately to a voiceover. But you can see in each of these, you should be able to find the 5-fold vertices where the big triangles, the 20, 1, 2, 3, 4, 5 of the 20 triangles come together in a point. And you should also see those triangles, the green outlined triangles, and in the middle of that triangle is a 3-fold axis of symmetry. And the slide, the projector slide, is at the 2-fold axis of symmetry because there's the same thing beyond the plane of the slide. Does that make sense? Okay, computer idiots, let's look at some more refined models. On the top left, this is rhinovirus, a 2-fold axis of symmetry. You could cut that thing right down the middle. It'd be the same on the right and on the left. Top right, smack in the middle of the triangle that's made by those 3 vertices, is a 3-fold axis of symmetry. And the center of the star, smack in the middle of the rhinovirus 14, common cold virus, that's appropriately oriented in the bottom one, is the 5-fold. Okay, so 2, 3, and 5-fold. And on the exam, I will have a virion, and I will have an arrow pointing to something, and I will ask you what that axis is. Okay, so be able to do this. Image reconstruction. So how do this works for smaller capsids? How do large viruses build solid capsids? So complex capsids, like that of herpes viruses, among others, can be built from several different kinds of capsid proteins that have similar structures So each of the proteins in the triangle can be the same, or they can look similar. They can have similar folds. Here, if you look in the second row down, that's a T = 3. So T = 1 is the simplest total number of subunits. 60 proteins interacting, the same protein interacting, and in the T = 3 virus, in that triangle, in that face, you see three molecules that are interacting somewhat differently. I'm gonna show you. And with this approach, better slide. So and this helps define a number which is complicated, called the triangulation number. It's the number of, this is what I was trying to say on the last slide, the number of structural units per triangular face. So in a T = 1 virus, it's the same structural unit that is making up that triangular face, and there are three of them. So 3 times 20 faces is the 60 units. Here's a T = 4 virus, where there are different proteins that are making up that face. and it helps to make a bigger. To visualize contrast, I've asked you to put together either a T equals 1 virus or a T equals 3 virus and bring it to class on Tuesday, and we'll take a look at them. That's that it. So here's the thing. I don't care that you can look at a complex virion and know that it is T equals 4, for example. But what I do want you to know is that 60 times T subunits forms, this is just sort of geometry, forms a closed shell with icosahedral symmetry. So canine parvovirus is a very simple virus. It's a small, T equals 1, and it has its capsid, this is a great table to look at to try to get it, has 60 copies of one protein called VP2. 60 copies of VP2. Picornaviruses, which we're going to talk a lot about, like poliovirus and rhinovirus, which you've seen pictures of, is a T equals 3. It has 180 different, it's made up of 180 subunits, and each one of those triangles has 3 major proteins, VP1, VP2, VP3, and hidden inside is VP4. So 60 copies of this is inherent in the part of those proteins. So here, let's take a look at the T equals 3 capsids of the coronaviruses. In 1985, the atomic structure of rhinovirus 14, common cold, and poliovirus were solved. And what was determined was that each of those proteins, VP1, VP2, and VP3, which make up the triangular face, actually had a similar structure. And so they might be different proteins, whereas canine parvovirus has 60 of the same protein. These have different proteins, but actually they're folded the same way so that they can pack against each other, frankly, as if they were the same protein, to pack together to assemble the icosahedron. And so one copy each of VP1, VP2, and VP3, which have the structure called a beta barrel jelly roll. And that's just the fold that protein makes. So samples, the beta barrel jelly roll turns out to be a useful fold of a protein evolutionarily to build an icosahedron. So beta barrel jelly roll domain proteins are found in plant RNA viruses, insect RNA viruses, DNA virus capsid proteins that have to fold to make an icosahedron. And what's mind-blowing is that proteins with completely unrelated primary sequences form the same three-dimensional structure. So evolutionarily, this is a geometry that allows icosahedral capsids to be built, and it helps explain how bigger viruses, which might use more different proteins, or smaller viruses, which may have evolved to use more different proteins, can solve the same geometric problem. Your frozen cryo. So here's the ribbon diagrams of VP1, VP2, and VP3 across the top. And while they make that structure, the beta barrel jelly roll, their primary sequences are different, and they have loops between some of the folds that pack together to form that sort of trapezoidy three-dimensional structure. And so those loops, or amino and carboxy terminal extensions of those proteins, turn out to be really, really important because they kind of decorate that beta barrel jelly roll. And the loops on the surface of VP1, one of those three proteins. Remember, VP1, VP2, and VP3 help to form the shell. VP4 is inside. VP1 loops form a cleft at the five-fold axis of symmetry, which results in a structure that kind of looks like a canyon. And so on the bottom right, you see like a topographical map of one of those 20 triangular faces, and VP1 at the five-fold vertex at the top helps to make a sort of rim for a canyon. And this will be important because the site at which the cellular attachment molecule binds, virus binds to the cellular attachment molecule, is in that canyon. So more on that later. Very low temperature. And then there's herpes viruses. And I think what's interesting about this herpes virus has an interesting structure. It's an enveloped virus, but it has an icosahedral capsid inside of it, and it has another layer in between the envelope and the capsid that is called the tegument. We'll talk about that quite a bit. But herpes viruses are big viruses. There are over 40 of the 80 herpes virus gene products are found in virion in different parts, many glycoproteins, tegument proteins. This is a large capsid. Despite the fact that it is big, a single protein, the major capsid protein VP5, forms the bulk of this T equals 16 capsid. There are other proteins that are found in smaller amounts, but basically the capsid is built from one protein, whereas the smaller pico, means small, coronavirus, its capsid is built from three major proteins to make the outside of the capsid, and one is found inside in equal molar amounts. So you can't just assume that because a virus is bigger, that more proteins are involved in building the icosahedron. Okay, does that make sense? Many micrographs are digitized. Okay, we're getting towards the end here. Again, this is an image of the structurally complex capsid of mammalian reoviruses. I told you earlier at the start of this structure section that three particle forms can be isolated biochemically that possess different functions. The virion on top is the environmentally stable form. The ISVP intermediate, or sometimes we refer to it as infectious subvirion particle, is missing the outer capsid protein sigma 3 and is prepared for membrane penetration. This is the membrane penetration active form. And then what's at the bottom of the diagram on the side is the transcriptionally active core. And you can see by cryo-EM structural differences. The open channel at the vertices in the core allow the product messenger RNA to be extruded, and inside I showed you a cutaway of where the molecular machine to the transcriptase is. The RNA-dependent RNA polymerase lies down there. And if we were together, I would do my interpretive dance of the reovirus ISVP to core transition. And if somebody asks me when we are together on Tuesday, I'll do the interpretive dance of the ISVP to core transition, but you have to ask. And aligned. Okay, the last image that I'm going to show is that of pox virus, so smallpox or vaccinia virus, and you can see that it doesn't really look anything like the others. not helical symmetry, not icosahedral symmetry, complex symmetry. And so, viruses have evolved lots of different ways of solving the problem, although it's not a problem, of being environmentally stable, yet disassembling appropriately what reaches the right place in the right time. Okay, the algorithm. So to end, I want to talk about the viral entry data sheet. It's due next Thursday before class, but you should start to fill it out as you're reading the chapter over the weekend. And so it would be good if you would start to read that chapter and fill in some information before we have class on Tuesday. Tuesday, I will talk quite a bit about viruses attaching to cells and entering cells. I'll get us primed, but then the details for influenza virus, picornaviruses, HIV, and reovirus, you're going to fill in as you come across them in the chapter, and you're going to write things in that table so that as a class we can pull the relevant facts together. And so I'll make sure that I post the slide set for Tuesday as soon as I can. I'm working on that now. So thanks for listening to this. I would really much rather, it turns out, be with you than doing this staring at my computer screen. But thanks for listening. And this, initially was always done with respect to icosahedral symmetry. So it was icosahedral viruses that could be analyzed using this approach because there is a structure around which the computer could orient the particles. The computer does the reconstruction and it applies the colors. Now this approach is actually getting more refined, and non-icosahedral viruses and particles can be analyzed in this way too.
Double-stranded RNA genomes, both of those need virion-associated RNA-dependent RNA polymerases in order to make messenger RNA. So making messenger RNA is important. Go down to number 4, double-stranded DNA typically uses in the nucleus, although not always, the cellular DNA-dependent RNA polymerase in order to make messenger RNA. And if you go all the way down to the bottom to number 6, you see, and we talked about this also, single-stranded positive sense RNA from a retrovirus is not infectious because this virus completes its life cycle by making a double-stranded DNA version of itself, and then that gets integrated into the host chromosome, and that requires reverse transcriptase. So that comes in the virion with the retrovirus.